Today's paper is the Triple Biology Foundation Paper 2 from 2023. Let's get stuck in. Hormones are important for controlling many processes in the human body. Hormones are produced by glands. Which organ system has glands that produce hormones? So the main thing to remember is that this is called the endocrine system. Again, if you're not sure about these, you can do a process of elimination, can't you? Circulatory system's all to do with blood, and nervous system is all to do with nerves. None of those have been mentioned so far. How are hormones transported around the body? Well, how is anything really transported around the body? Number one way of getting anything around is in the bloodstream. Figure 1 shows glands in a woman's body. You've got these A, B, C and D labelled, and then ovary, and then it's matching them up. So again, this is another one where it's just a, can we remember it? So up in here in the brain, we have the pituitary gland. This is often referred to as the master gland because it controls a lot of the processes. B here is the thyroid, responsible for hormone growth, human growth hormone. My dad's thyroid's underactive, lots of people's are, so they have to take tablets to improve that. These here, which are kind of on the top of the kidneys would live here. Okay, those are the adrenal glands. And sometimes you get really kind of, I don't know, you get spooks. You can feel that adrenaline almost coming out of the back. You almost feel it pulsing from there. Or maybe that's just me. And then D, which isn't actually one of the ones of the options down below, is the pancreas. And remember, the pancreas, as well as producing enzymes in the digestive system, is also there for insulin. So, given what we've said, this should be pretty straightforward. Just remember that when it says draw one line, it comes from the left, right? Here you've got 4 options. That does not mean that you have 2 lines coming from one of them, so each one of these has 1 line. It is 1 line from this lot. I know it, many of you will think, you know, that's obvious, but you'd be surprised how many times that happens. Again, you could also see, look. Right, 1.4, which gland in figure 1 produces insulin? So we've already spoken about this. D is the pancreas. Which organ does insulin mainly affect? So if insulin, if you remember, is there to take our blood sugar, our excess blood glucose, and we turn it into our store, which is glycogen. And this happens, and this is stored in the liver. So it is the liver. Give one effective insulin. We've already done that there, look. See, look, it's almost like I know what's going on. So one effect is it reduces the blood glucose level by storing the glucose as glycogen. So you can always talk about that as converting. Some hormones control a woman's menstrual cycle. Which hormone causes an... Thanks for that up top. Some hormones control a woman's menstrual cycle. Which hormone causes an egg to mature in the ovary? So, you have got, let's use this as an opportunity for a little bit of revision. There are kind of 4 main hormones that we need to think about. So we've got LH, FSH, estrogen, and progesterone. And the menstrual cycle is a really good example of how the hormones all interact and work with each other. Okay, so the pituitary gland, so this is made in the pituitary, makes FSH, and as you can see up here, it's follicle stimulating hormone. And what that does is it does 2 things. Okay, is it starts an egg being matured, okay, and it kind of turns on estrogen. Okay, they all are going to interlink. The estrogen, which is made in the ovaries, what this is going to do is this is going to thicken the womb lining. Getting it ready for implantation. But also, it stops, maybe I should have done that in a different colour. Yeah, let's do that. It stops FSH production. So like this is turning it on and then that's turning it back off, all right? Now LH, okay, LH has one job basically, and that is to cause ovulation. So the egg to be released. So you get this massive peak of it around day 14, okay? Now progesterone, you can think of as kind of I always think about it as our pregnancy hormone. What that does is it tells the womb lining to kind of, it maintains it. And while that's doing that, that kind of, again, inhibits a little bit of that, should have done that in orange, inhibits a little bit of that FSH coming on, because it's like, hold, we might be pregnant, hold, all right? So it keeps that maintained. It also kind of thickens the mucus at the opening of the cervix. So I know I'm kind of going out of order here, but you can see just straight above, question 1.9 says contraception prevents pregnancy. Give 2 methods of contraception that use hormones. So you've got the contraceptive pill. And what hormones would I use for that? I'd use estrogen and progesterone because they stop the FSH making, so you don't make any new eggs. And also kind of by taking the progesterone, your body's almost thinking it is pregnant. Okay, so that's, I'm just adding that in because, you know, I'm a fancy man. That use hormones. So you've got a couple of options here. You can think of an IUD, a coil, an intrauterine device. You can think of an implant. Okay, there's patches, all sorts of different things. So going back originally to here. Which hormone causes an egg to mature in the ovary? So we've just spoken about that. That is our FSH. Which 2 hormones help maintain the lining? Estrogen and progesterone. Again, even if you're not sure, these are the only ones that we've mentioned which are to do with the menstrual cycle. Amylase is an enzyme, right? That's what gives it away as the A's. Same here for protease. And thyroxine is a hormone. But if you look at the word, right, that is thyroid. That's where that's made. So there's nothing to do with that. Okay, moving on to question 2. Students estimated the population of buttercup plants grown on a lawn. You've got a rectangle measuring 15 by 10. This is the method used. They measure the length and width of the lawn, choose 5 locations to sample, place a 1 by 1 square frame. Yes, boys, we love a quadrat. Record the number of buttercup plants in each square frame. So first one is just using some equipment. Length and width should be measured using, well, you're not going to do it using a 15 centimetre ruler. You'd use that using a 30 metre tape measure. Oh, look at it. Look, I'm just hard coded. It's called a quadrat. Lovely. How should the students choose it? They should do it randomly. Okay, so there's a couple of ways of doing this. You can like mark out the sections. The way you've probably done it at school is because your teacher was like me, just feeling a little bit lazy, just said go out on the field and huff it, right? And you absolutely lob it somewhere, and where it lands, you record it. But what you can do is you can kind of divvy it up and like number the bits, and then roll dice or some kind of random number generator, and that then tells you where to put it. Table 1 shows the results, so 2, 7, 0, and 1, lovely. And they use the results to calculate the population. So if you multiply the length of the lawn by the width, that will give us the area. Hopefully we're okay with that. And the total number of buttercup plants and divide by 5 to give the mean. Remember, that is kind of what these are all, so median is also a type of average, but I would say if you think about mean, if you ask most people what an average is, they'd probably say the mean. The students calculated the population of buttercup plants on the lawn was 300. How did the student use the results in table 1 to calculate the population? So what they do is you take the mean, because that tells you how many you expect in 1 by 1 meter. You can then take the area, and that will give you the volume. That's not the volume. And that will give you the estimate of the population. Okay, how could students improve the accuracy of the estimate? Well, if you think about it, right, so this defeats the point because that's not random. Using the square frame measuring 0.05, it's just going to proportionally kind of do the same vibe. You're going to need to do more, right? If you have more samples, you're going to have a more accurate measurement. Because I suppose, if anything, if you think about it, right, the most accurate would be you literally count all the flowers, and that would be sampling every area, which is stupid, but like you can see how that works, right? The closer you get to sampling everywhere, the more accurate you're going to be. 1 by abiotic. So abiotic means not living. And a biotic factor is one which is living. So these are things which affect the population of things. So if sunlight, for instance, is not living, and that would be what I would put as my thing. But other things you could also have, things like temperature, things like water, things like the wind, minerals. All these things are not living, and they're present, and the plants are relying on them to live. So biotic things might be if you've got a sheep on the field who likes to eat them, right? So predators in a way. Yeah, it's a non-living factor. Right, different species are in a habitat may depend on each other for food. Here we've got a food chain. The grass needs energy to grow. What is the source of energy for the grass? So again, ultimately that is coming from the sun. However, okay, sometimes they like to be a little bit picky and want you to say light, but sun is fine. We've got this table of different feeding relationships. So let's label them on here, because then I think that makes it easier to fill in our table. So the grass is the producer, because it is what is first taking that energy into the form of biomass. If you are eating a producer, you are a consumer, but because you're the first thing eating another living thing, so if you eat another living thing, you're a consumer. If you're the first one, you are a primary consumer. And then just like you have primary school, you then have secondary school. So the lynx is our secondary consumer because it eats the primary consumer. Let's fill them in. So our primary consumer is our hare. We've got our grass. Now, a herbivore only eats plants. And you'll notice the only thing which only eats plants here is the hare. The carnivore eats meat. So the lynx will be eating the hare, so therefore it's the lynx. The prey animal is one which is eaten, so that's the hare. And the predator is the one which does the eating, and that is the lynx. Okay, just be aware, obviously I know it's obvious here because there's 6 things and you've only got 3 that you can be using more than once, but it would always mention if you can use it more than once, and very often people don't look at that. Right, the appearance of the snowshoe hare in the summer and in the winter. So there it is in the summer, there it is in the winter. The snowshoe hare has a different fur colour in the summer and the winter. Explain how the different fur colour increases the chances of survival of the snowshoe hare. So we're looking really here at adaptations, things that animals do to survive longer. And you'll notice here this is an explain question. So that means I'm saying why. Why is this the case? So I think the first observation that you've got here is, and even printing it in black and white is even better, right? Because you can see here that it just blends in, all right? So the fur colours are camouflaged for the season. Okay, brown and thing. Anything to do with blending in surroundings, anything like that would work well. You do not have to, I think it's really easy, especially I know you've now finished English as of time of recording, but there's a lot of things thinking that you need to have this really like elaborate written answer. No, just bullet points, it's fine. Fur colours are camouflage for the season, right? So then we're explaining why. Why is that relevant? That means they're less likely to be seen by the predator, or which in this case is the lynx. And if they're less likely to be seen, they are therefore less likely to be killed or eaten. Okay, often with an explain question, you've got one kind of insight and then it's what does that mean? What does that mean? And you can see how that mirrors through there. Right. Figure 5 shows the number of snowshoe hares and the number of lynx varied in one area. And you can see you get this kind of cycle effect. Figure 5 shows the number of snowshoe hares and the number of lynx increase and decrease several times. Suggests 2 reasons why the number of snowshoe hares increases. So, why would the number go up? All right. So, if I kind of go back to that food chain, why would the number of this go up? Well, it's got to be for 2 reasons. It's either that we have more grass or that we have fewer lynx. Because if I've got more grass, I've got more food, therefore I can have more snowshoe hares. If I have fewer lynx, that means less snowshoe hares can be eaten, so the number stays higher. So you kind of get this pressure from each end of the food chain. If you have fewer of the animals above, you're going to have more. If you have more of the animals below, you've got more food, and therefore you can have more. So more grass or fewer lynx. Okay, the number of snowshoe hares increased and decreased 4 times between 1900 and 1935. What effect does an increase in the number of snowshoe hares have on the number of lynx? So, if I have more hares, if I go back to our thing, if I have more hares, right, so let's do it in a different colour so it's obvious. If this has gone up, that means that lynx have now got more food, so that goes up. And this is why, when you look at this, graph, you get this cycle, because you can see here that the hares increase, and then a little bit below, a little bit after, the lynx have now got more food, so they increase. And because there's more of them, that means that the snowshoe hares decrease, and therefore the lynx has decreased. So you get this kind of like effect where the only way I could easily describe it is if you ever did this when you were younger with like your colouring pens and you start doing like this. That's kind of the shape that you get. Right. Recover the pen lid, which is flung across. So increasing number of snowshoe hares will increase the number of lynx. That's enough. So just one reason why the lynx decreased from 1915 to 1919. So again, can you see here, right? So 1915 and 1919 is like here, right? Look at the snowshoe hare level, low. So therefore, the lynx is going to go down. It is not some dramatic thing where like there was a load of lynx fighting in World War I or something like that. although I would quite like that. That'd be quite impressive. So there is a very low number of hares to eat. When the snowshoe hare eats grass, about 90% of the biomass of the grass is lost. Suggest 2 ways that this biomass is lost. So what this is essentially saying is, what do we do with our food? What do we do with the energy? What do we do with the food? So some of it will go to growth, and this is the stuff which is maintained, right? So the rest of it has got to go to things like heat, things like, well, the heat is coming from, you're doing respiration with it for energy. You also, let me change that to food, actually, it's probably a little bit better. You also don't absorb all your food, so things like feces, poo, that is all of those. You don't eat it all, right? You're not eating the bone, all these types of things. So the first thing, the only thing you can't say is growth, because that is going into our biomass. So the first way I would say is that you are using it for respiration, and that you make feces. On to question 4, powering through. Some farmers keep cow indoors in large shed. Others farmers keep cows outdoors in fields of grass. Look at that. Look at that. It's like a Windows backdrop. Figure 6 shows cows being kept indoors and outdoors. Table 3 shows energy inputs for keeping cows. So the input is food. So this is how much you're giving them. Input is fossil fuel, because, you know, in the indoor one, you're going to want to heat up the thing. And how much output you get as meat and milk, which first of all is a walking advertisement for being a vegetarian, because look at all that energy you put in and how much you get out. It's all right, I still like meat. Calculate the total energy input for keeping cows outdoors. So these are our inputs. So we've got 5950 in the form of food and 50 in the form of fossil fuel. So that gives us 6,000 kilojoules per metre square per year. Next one, total energy input for keeping cows indoors is 16,000. Calculate the percentage efficiency for keeping cows indoors. So again, you might be thinking, this isn't physics, but it doesn't matter, it's given it to you, put it in that thing. Also, you're allowed to kind of bridge the gap a little bit. So energy output here is 40. It's given you that it's 16,000. So again, we always like to follow FIFA. So we always put our formula, write our formula. That's already given to you, so it's not as problematic here. Insert numbers. We then fix that if it's necessary, and we get the answer. So we're just going to stick our numbers in. So our percentage efficiency is our output. What we get out is 40. Our input is 16,000 times by 100. I don't need to do any kind of fixing here. Everything is in the right units. Everything is in the right format where I can just put it into a calculator. So therefore, we do exactly that. And our efficiency is going to be 40 divided by 1,600 times by 100, which I believe is very small. Off the top of my head, that'll be, I think it's 0.25%. Okay, lovely stuff. Bit of mental maths. Right, the percentage efficiency for keeping cows outdoors. By the way, I know I said about mental maths, stick it into a calculator. No, don't faff around. The percentage efficiency of keeping cows outdoors is 0.03%. Why is it more energy efficient to keep cows indoors than it is outdoors? So, the number one stupid mistake that people do is not ticking the number of boxes that it asks. I know it sounds obvious, but like I've said to lots of people this year, like if you imagine with like 5 minutes left in the exam that I come in and just sanity check, right, go back through have you ticked 2 boxes? Have you done 1 line? All of those things. So cows are more stressed indoors. That's not a reason to keep them indoors. Cows move less. That's the big thing, right? If they move less, we're going to need less energy for respiration, so more of that energy can go to growth. Again, if it's warmer, I need less energy for that respiration. I can go to growth. All right, so that's the idea. If you like, this is why battery farms are really efficient, but I mean, they're horrible from a welfare point of view, but you're not making the move. They don't have to waste any energy. They can just get fat, basically. Diseases in cows cause problems for farmers. Suggests why diseases spread more quickly when the cows are kept indoors. Because they're really close together, right? And if I have, if I have a sniffle when I go into a crowded classroom, people are probably going to get sniffles. One species of bacterium causes a disease in cows. Scientists investigated the effect of 8 different antibiotics on the growth of this species. The scientists put discs containing the different antibiotics onto a petri dish, and you can see you've got A to H. The species of bacterium is resistant to some of the antibiotics. Give a letter of one antibiotic the bacterium is resistant to. So again, you might be thinking, I was done there with this. This was infection response, but it's because it's going to go on and talk about the idea of developing it and the resistance from a kind of an evolution point of view, I suppose. Now, if you remember, the area around indicates that it has killed the bacteria. So if I have no area around, that means it hasn't worked. So A has worked, B has worked, C has worked very well. D has not worked. G has not worked, and H has not worked. So those 3 have got nothing on them. Either one of those works as an answer. Antibiotic resistance in a single bacterium is caused by a change in the bacterium's DNA. And this is where it's kind of now relating to paper 2. Okay, but DNA are a molecule which encodes for everything that makes you, makes everything a bacterium, so on and so forth. A change in one bacterium can cause millions of bacteria to become resistant to the antibiotics. This is because bacteria have a high rate of, going to the toilet's not going to matter, is it? Eating things is not going to matter. It's because if I have that trait appear in one bacteria, it will very quickly become 2, very quickly become 4, 8, so on. So it is because it has a high rate of reproduction. Suggest why the production of millions of antibiotic-resistant bacteria is a problem for farmers. So if I have loads of antibiotic-resistant bacteria, that means that I'm going to have more of my animals become ill. And if I am ill, right, what happens to you when you get the flu? You lose weight and you kind of feel a bit weak. Same thing for animals, right? So if they are ill, then they are going to be less kind of efficient, as it were, at producing meat or dairy or whatever. Okay, and therefore, ultimately, it comes down to money, right? Jeremy Clarkson will go and cry in a field, right, with all of the thing. In fact, actually, if you have watched Clarkson's farm, when he has to put down his cows, he does actually go and cry because, that tuberculosis has meant that he has to cull his cows. So that ultimately impacts the farmer because the animal's not going to be producing as much meat. Question 5. Bacteria are one type of organism that cause decay. Which other type of organism causes decay? The big one here is fungi. Students investigated the effect of temperature on the decay of milk. The decay was caused by bacteria in the milk. Right, let's just straight up. I know we're about to launch into the investigation, but let's think about it, right? This is sometimes worthwhile, is just think, what do I know, right? What do I do with milk? It goes in the fridge. Why does it live in the fridge? Because I don't want it to go off. If you leave the milk out, right, then it starts to spoil and go off. So I'm going into this expecting a lower temperature means that you're going to have lower decay, because that's what we do in real life. So this is the method used. They've got the fresh milk, the thermometer, and the pH meter. And again, if you notice, one of the ways you tell that milk has gone sour is it starts to taste like one of the ways that you tell that the milk has gone off is it starts to taste sour. The pH sensor will show that the pH is falling, becoming more acid, and that means it's gone off. So set up the apparatus, record the pH each day for 7 days, and repeat it with samples at 5 degrees and at 30 degrees. What was the dependent variable? So the way I always like to remember this is dairy milk ice cream. So the dependent is the thing that you measure, and the independent is the thing that you change. So if we're looking for the dependent, that means the thing that we're measuring. And what we're measuring in this setup is the pH. How could the students keep the milk at 30 degrees for 7 days? So if you've done a temperature experiment in class, which you should have done with the enzymes, is you can put that in a water bath. It's like a giant kind of, it's a bath, it's kind of in the name, isn't it? And the water can be kept at 30 degrees, and then I can put my milk in there like that. Lovely stuff. As the milk decays, the bacteria digests fats in the milk. What type of acid is produced by the digestion of fats? Don't overthink it. It is fatty acids. Amino acids come from proteins, and hydrochloric acid is your stomach acid. And fatty acids and glycerol is what is produced from the digestion of fats. Figure 9 shows these results. Why did the pH not fall below 4.5 at 20 degrees? So for 30 and 20, you can see you kind of max out at 4.5. Just like when you do rates in chemistry, that kind of flat line means that the reaction has stopped, right? It means it's all reacted, it's all broken down. The digestion of fat was fastest at 30 and slowest at 5. Give one reason why the rate of digestion was faster at 30 than at 5. Well, again, you don't want to feel like you're straying too much into chemistry, but if at 30, okay, there is kind of more energy. At 30 degrees, they're moving faster. You could also say that the enzyme works better or has works faster. Sorry, so it's like better activity. You could also say that bacteria, because I mentioned bacteria were there, you could say that they were dividing faster as well. Calculate the rate of digestion at 30 from day 2 to day 3. So it's held your hands to it, but I always like to show my working out when it comes to graphs. So if it says day 2, we're going to go up from day 2, and we're looking at the 30 degree one. So we're going to go up here and across, and you'll see that the pH of that is 6. At day 3, and I read across, that is 4.7. So the fall over that one day, it's gone from 6 to 4.7, not quite 6.7, is 1.3. The rate of digestion at 5 degrees from day 2 to 3 is 0.1 pH units per day. How many times faster is the rate of digestion at 30 than at 5? Again, it says use your answer to question 5.7. So first of all, if you did muck that up, as long as you use that, you get something called error carried forward, so you'd be fine anyway. But gives us as 0.1, so notice that that's 13 times bigger. So if it's 13 times bigger, that suggests it's 13 times faster. Okay, the kidneys filter the blood. Our triple only stuff here. You can see here I've got like kind of the filter in the kidney. You've got the blood. You've got water in there, sodium in there, urea, glucose, protein. It says glucose molecules are found in the blood plasma and in the filtrate. Protein molecules are only found in the blood plasma. So if you have a look here, right, they've got a fit through here. If I look at that protein, no, that is not happening. So the reason that the protein is not in the filtrate but is in the plasma is that they are too large. And for the glucose, the reason they're in both is because, again, they are small enough to pass through. You shouldn't ordinarily have glucose in your urine. That's actually a sign of diabetes. But again, remember, one line. It doesn't mean from the reasons, it doesn't mean from the right, it means from the left. The kidneys control the volume of water in the body. Table 4 shows information about a person on one day. So we've got the water filtered from the blood and the water lost in the urine. Calculate the volume of water reabsorbed in the blood. So if I have got, right, 160 through to this point, and then that then goes into our urine, and you only get 1.9 there, that means that quite a lot of that has gone back, right? It is just a subtraction. 160 minus 1.9 gives 158.1. It's 1 mark, so that means we're not expecting fireworks here. Okay. Oh, now this is where some of you mentally give up. You're like, oh, look at all those lines, I ain't dealing with that. We got this. A person with kidney disease may be treated by dialysis or having a kidney transplant. They love this type of question. Figure 11 gives information about dialysis and kidney transplants. So we've got all this stuff here. And again, remember, they always, examiners like to think they're interesting. So what they do is they make the questions relevant, but that means that because that stuff is not in the specification, they have to give you a load of information. So for dialysis, we've got a person needs 3 dialysis sessions a week, each session lasting more than 8 hours. Most patients have dialysis in hospital. Protein and salt levels in food must be kept low. Dialysis costs £35,000 per year. And we've got the same thing here. We'll come back to it because we are going to evaluate them in a second. A doctor states it is better to treat a person with kidney disease by using a kidney transplant rather than by dialysis. Evaluate the doctor's statement. Evaluate means you have to compare both sides and you need to give a conclusion. Use information for figure 11. So, if I want to look at both sides, that means I need to look at, and I want to give a conclusion. That means I want to look at why transplant is good. I want to look why dialysis is good. And then I need to say overall. So this is going to be a 3-paragraph answer. So we're going to use 3 colours. Let's look over here. So a person needs 3 dialysis sessions a week, each session lasting more than 8 hours. Okay. Whereas here, the transplanted kidney will work well for about 10 years. Okay. So you've done it. It's 1 operation and you're done. So this is a bad thing for dialysis, right? I'm going to put the arrow. I'll tell you what. Let's say if it's red, it's a victory for transplant, right? And if it is orange, that's a victory for dialysis. Most patients have dialysis in hospital. You have to be in hospital, that's not good. Protein and salt levels in food must be kept low. Well, I want to eat what I want. Dialysis costs £35,000 per year for each patient. If you have a look here, transplants cost 17,000. So, oh my goodness, it's like an absolute romp for the transplant at the moment. In a surgical operation, the use of general anaesthetic can usually cause damage to other organs. Well, that's a win for dialysis, right? Because there's a risk associated with the operation. After a transplant, the patient must take drugs for the rest of their lives to suppress their immune system. That's pro-dialysis. The transplant costs £17,000 and £5,000 in each of the following years for drugs. Okay, so that's a win for the transplant. And then the transplanted kidney will work well for about 10 years. So it depends on how you want to argue this, right? It's 10 years, that's 10 years that you wouldn't have had otherwise, but it's not forever, okay? So let's kind of do that as a draw, right? Now, there are some other things. Is there any other bits that we know? Okay. Any other business? Right. First of all, when you've got a transplant, you have to wait for an organ. Right? Dialysis is a machine. Why do we need to take those drugs? That's because we can get rejection. Okay, that's where your body says no. You've got the other risks with surgery. So with surgery, you've got things like infection or bleeding out, right? This is a major surgery. As much as the dialysis is inconvenient, okay, as I said, you don't have the supply issues of a transplant, okay? Now, it's really important in our answer that we're not just regurgitating these points, we're doing something with it. So we're not just saying most patients have dialysis in hospital, we're saying why we think that that's worse, right? We're saying, so therefore you have to have inconvenience or you're having to spend time in hospital, right? If you just reiterate the points, you're not going to get anywhere. So it's about doing something with it. Let's actually write the answer, shall we? So the first paragraph, why is a transplant better? Okay. So the transplant does not need regular sessions or visits or dietary restrictions. Why is that bad? So why is transplant better? So you have less lifestyle impacts, right? What we're saying here is the transplant, because it doesn't need regular sessions or visits or dietary restrictions, your life is not impacted in the same way. Okay, the transplant is cheaper. Let's use the numbers. By £18,000 in the first year and 30,000 each other year. Okay. We haven't actually mentioned as well that so dialysis is like a giant machine where the blood comes out of you and then goes back in. So you have all the problems with the machine, okay? Doesn't have the risks with dialysis, which is you get clots, you can get bloating, because basically until you have dialysis, your body starts to swell up. If you know Spudman, the guy who sells like the jack of potatoes and stuff, he's on dialysis, right? So he'll often say about how he's like really tired and he has to set up his dialysis at home and all sorts of different things. It's really restrictive. It doesn't have the risk of dialysis like clotting, bloating, okay, and other impacts. So let's look at why dialysis could be better than a transplant. Okay, there is no risk. So dialysis doesn't need to wait for an organ. Okay. There is no need to take anti-rejection drugs. Or have a risk of rejection. Okay, the transplant is a major surgery. With risks of bleeding and organ damage, as we saw up here. Okay. It might need replacement after 10 years. So it's not like a fix all forever. So then we have to say overall, what do we think? Okay. And overall, and then you can agree or disagree. I would say personally, okay, I agree that transplant is better as it enables people to live full, normal lives. Right. Next question. A kidney transplant costs £17,000 in the first year, then £5,000 in each of the following year. Calculate the cost of the kidney transplant over the first 5 years. So year one, we're going to have 17,000, and then year two is 5, 5 in year 3, 5 in year 4, 5 in year 5. Adding that up is £37,000. Lovely stuff. Okay, many species can live together in the same habitat. Lovely. What name is given to all the organisms living in the same habitat? Okay, so if you are all of the organisms is the community. The population is all the organisms of one species. Okay, and an ecosystem is the community and the habitat mixed together. So that is like kind of all the animals and where they live. We call that the ecosystem. Food chain is what we've seen before. Figure 12 shows four species of bird from the same habitat in the UK. Got a brambling, bullfinch, a chaffinch, and a goldfinch. Which species of bird do they think are most closely related? So, I think I've seen people before try and do this by like looking at them, but this is really a question on classification. So if you remember with the Latin names, okay. Oh. If you remember with the Latin names, you have the last two are genus and species. So obviously, if you are the same species, you are really closely related. The next level up is genus. So if that's the same, that means you're kind of in the same family. Kind of like a lion and a leopard, right? You aren't there in the same, like a lion and a tiger, they're in the same genus. So they can have non-fertile offspring. If I have a look here, fringilla, so that means it's the brambling and the chaffinch, because they are both in the same genus. Scientists think that the brambling and the bullfinch belong to different species. Bum bum. What evidence is used by scientists to classify the brambling and bullfinch as different species? So the number one thing is what this is really saying is what is a species, right? If you're saying what evidence is that they're not a species, this is really a question about what is a species. And we've kind of hinted at it when I was talking about a lion and a tiger. The idea of species is that they can have fertile offspring. That means they can have babies, and those babies can have babies. So the brambling and bullfinch cannot breed together to give fertile offspring, which means they're not species. They're not in the same species. 4 other species of bird live in a habitat in the UK. Figure 13 shows the numbers of each species of bird varied during 1 year. Describe what happens to the number of birds of species A through the year. So it's 3 marks. So when you're describing a trend and it's 3 marks, that means there's going to be 3 kind of sections. Well, let's have a look. Here, I would say is section 1. All right. So the number is constant and low from January to kind of April. Right? Now when I look, next kind of thing is we get this explosion up to here. Okay, so we get a rapid increase April to May. Okay, and then we have a gradual decrease for the rest of the year. Right, there's 3 sections, 3 marks, nice and straightforward. In June and July, the disease affected the populations of some of the species. Which species had the lowest resistance to the disease? So, in June and July, so let's look at here, we have a disease, and you can see that you're getting this kind of marked down. So if I have the lowest resistance to disease, that means I'm going to have the biggest drop, all right, because most of them are going to die. And if you have a look here, this one has got the biggest decrease. That therefore presents it as B. One species migrates between the UK and other countries. Which species migrates? So if it migrates, that means there's sections of the year where it's not here. And if you have a look, that to me looks like D, because there's 0 of them here. Then the weather gets sunny, they've come back and they've gone like that. So it's D. The reason is that there are no birds during autumn and winter. Okay. Question 8. Getting there. A person's eyes can focus on objects at different distances. A person looks at a distant object. The person then looks at a near object. The person's eyes make adjustments so that the near objects form a clear image. Which term describes the adjustments of focus from a distant object to a near object? Okay, so these ones here are actually diseases to do with the eye, right? They are basically short-sighted and long-sightedness. Adaptation, I can understand why you might want to go with that because you are adapted it, but adaptation is more of a wholesale thing, like having eyes or having particularly good eyes. The changing adjustment of the focus is called accommodation. Which structure in figure 14 is where the image is focused? So the light comes in here, is adjusted by the lens and is focused on the back of the eye. That is B, and that is the retina. Which structure in figure 14 contains a muscle that contracts when focusing on a near object? So you can see here how you've got the muscles, and what that will do is that will is you can stretch and squash, and that will change the size of the lens and therefore enable it to focus at different distances. So that is structure E. What happens when it's focusing on a near object? So if I have a very thin lens, right, okay, then it might bend it, say that much. If I make that lens fatter, right, it bends it more, all right? If I have got an object really close, because the light is kind of is spreading out rather than coming from parallel lines, you actually need to focus it more. So the lens gets shorter / fatter, however you want to word it. The eyes can function in dimly lit areas and in brightly lit areas. The iris contains muscles. Describe how muscles in the iris help a person see clearly when moving from a dimly lit area to a brightly lit area. So your teacher might have got you to do this. If you haven't, it's quite a good thing to do, where you look into someone's eyes, very romantic, and then you turn the lights on in the room and you see that their iris will shrink so that it restricts the amount of light entering your eyes so it doesn't overwhelm you. So in a dimly lit area, okay, the iris is relaxed and pupil is wide. Okay. And so when it goes to into a brightly lit area, okay, the muscles will contract, okay, the pupil will shrink, slash get smaller, and less light will enter. It is important to be able to react quickly. Think fast. Many people think that drinking coffee decreases reaction time. Plan an investigation to test the effect of drinking coffee on reaction time. So we've got our required practical question here, the reaction time practical. You should include the test for reaction time that you would use and how to make the investigation valid. So the practical that you might have done for reaction time is the ruler drop, where you have your ruler, you have your person with their like finger and thumb there ready. Look at that, what on earth is going on there? They drop it, you catch it, you see how far it is. Okay, so if we are looking at the effect of drinking coffee, again, we think of DMIC. We're wanting to measure the dependent variable. The one that we want to measure is our reaction time. So how are we doing that? That will do our ruler drop distance. That's what that's going to be. And the independent, what we're going to change each time? Well, we want to see the effect of drinking coffee. So that means that we need to change the level of caffeine or coffee. Okay. So let's set up our experiment. Okay, let's do the experiment and then we can introduce our independent variable afterwards. Okay, whenever we're doing an experiment, remember we want to make sure that we're making it nice and clear. We can use a diagram, we can use bullet point lists. That's what we can do. So number one, okay, is we are going to have position a ruler above the finger and thumb, release the ruler randomly and have the person catch it, record the distance of the drop. That's the fundamental practical. So that is really tick done. But we need to make the investigation valid and we still need to think about how we're doing that there. So then what we would do is we can say, right, repeat the experiment 3 times and calculate a mean. And then, crucially, so we're doing the effect of drinking coffee, we then repeat after drinking a cup of coffee. All right, now, if I want to make it valid, I need to also think about what my control variables are. So our control variables are things that we keep the same. and that is so it is fair. So for it to be valid, okay, ensure that we control for, and we can do a bit of a list if we want, we could have the same age, right? Your reaction time might get worse when you get older. Same level of sleep. If someone's well rested and someone's jet lagged, it's going to be a problem. We want to use the same coffee and the same volume of coffee. And we have the same time interval between drinking and testing. Because, you know, if someone's necked a coffee and then waits 8 hours doing it, is it actually going to be valid? Okay, lovely experiment. Right, 2 more pages, last question. Reproduction can produce offspring which are genetically different and genetically identical. Farmers grow tomato plants in greenhouses. Tomatoes are sold in supermarkets. Suggest one advantage of growing tomato plants that are genetically different. So what this is really hinting at is what's the point of variation. Okay, so the good thing about variation is that not all of the plants might be susceptible to a disease. Okay, and you also might get some nice new tastings. You might get new flavors. Okay, but that top one is really important. You've probably heard of the Irish potato famine. Okay, I mean, there's lots of reasons why that happened, but fundamentally, you know, there was potato blight, and that was a disease which affects potatoes. It wipes them all out. If you have genetic variation, then not all your plants might die from a disease. So if all your tomato plants are identical and then there's a disease which comes out which kills tomato plants, good luck. Suggest one advantage of growing tomato plants that are genetically identical. So if you've got a really nice tasting one, so like, you know, think of apples, right? You can probably name all these different types of apples. They are all clones of each other because we know that, you know, Pink Lady apples taste nice and do this. So therefore, why we don't want to change it, okay? So it means that all will have the desired taste / characteristic. Okay, they'd all, you know, they'd all be ready at the same time, all of these types of things. Scientists can grow genetically identical tomato plants using tissue culture. What is tissue culture? So this is where you take some cells, tissue from the plant, and then you grow that into a new organism. So it is cells from the organism are taken and grown into a new one. Genetically identical tomato plants growing in the same garden do not all grow to the same height. Okay, so give one reason why. So this is really thinking about what other factors are going to affect growth. So if you're in one part of the garden, you might have different amounts of sunlight. You might have different water. You might have different minerals, like one part of the soil might be rubbish. All these different types of things. The sex of dogs is determined by X and Y chromosomes in the same way as humans. Complete the Punnett square diagram in figure 15 to show the inheritance of sex in dogs. Use the symbols X and Y. So in humans, broadly, female is XX. So that means that the gametes could either be an X chromosome or an X chromosome. And XY for male. So we then look at see what would happen when these, if I remember, because remember the gametes are produced randomly, so I could produce an X or an X, an X or a Y. So if I got this X and this X, that make XX, which is a female. Okay, XX, which is a female, XY, which is a male, and XY, which is a male. A female dog gave birth to 6 offspring. Why would you expect there to be 3 male offspring and 3 female offspring? Use your answer. Okay, so there is a 50-50 probability of boy to girl or male to female. Okay, so they're like equally likely. Okay, and yeah, therefore if they had 6 offspring, you'd expect 50% of them to be boy, 50% of them to be girl. Lots of farming in this paper. Farmers keep chickens for meat production and egg production. Some varieties of chicken grow more quickly and are more suitable to meat production. Other varieties of chicken produce more eggs. So you can see this one grows quicker. This one has more eggs. Don't like this graph though. It doesn't start 0. Oh. Other varieties. Oh, that one shocking. Other varieties of chicken produce more eggs. They keep the varieties. Farmer investigate the growth rates and egg production of both varieties. Here's the results. Suggest 2 control variables. Okay, so if I'm just wanting to see how many eggs they are, right, I want to make sure that I'm giving them the same type / amount of food, because if I'm if I'm feeding one more, that's unfair. We've experienced earlier in the paper about if they are outdoors or indoors. I want to make sure that they're all doing the same there. Things like temperature you could have, like the amount of space, the time of the year, right? I suppose in theory you could say you've got to make sure that they have the same mass at the start. Lots of different things. Figure 16 shows mean values from 500 chickens of each variety. Give the reason the farmer used a large variety of chickens. So if you have a large number, that means that your answer will be more valid or representative. Because if I'm asking more people their opinions, then if I asked everyone in the country their opinion, I suppose what you do on a poll, isn't it? Then that's the most accurate. But if I ask as many as people, I'm going to get the most kind of comparable amount. You could also say it provides the most accurate mean. That's another thing you could say. The farmer wants to produce a variety of chicken that is good for both meat production and egg production. Describe how selective breeding of chicken varieties A and B can produce the new variety of chicken. So, selective breeding is basically evolution on steroids. So it's not actually, it's probably a poor form because sometimes they do use steroids. But we've got this idea of variation. And in evolution, it's selected by which one is better adapted. But in selective breeding, you're doing the selection. So what we'll do is, the first thing that you'd do is you'd have A and B breed together. Okay. And then what you do is you would select the offspring with best egg and meat production. And then what you would do is you make them breed. Okay? And then what you do is you'd select the best again and repeat. And eventually you should get your desired chicken. Okay, so over subsequent generations, it all kind of clicks together. Right, quite a long one there, but that is the Biology Paper 2 Foundation 2023. Hope that helps.